Is birth a source of food security or food sovereignty? For the purpose of this discussion, I want to differentiate between what is food security and what is food sovereignty. Food security is a situation whereby you get enough food, either is of quality or is of quantity, as long as you have it, and wherever it doesn't address where you get that food from. But when you are looking into food sovereignty is a situation whereby you produce locally what you are eating, and excess of it may be taken to another environment or to export it to another country. So, for the purpose of that, I'm going to look into, do we need food security or we need food sovereignty? Then from there, we look into the concept of destructive technology and food insecurity. Lastly, is the issue of sustainable 1, which is no poverty, sustainable 2, zero hunger, sustainable 13, climate action, and lastly, life on land, which is SDG 15. Now, in social sciences, unlike in physical science, we cannot use only one theory in order to look into our problem, because we study human behavior. And when you are looking into human behavior, there are so many variables that determine your behavior. It starts from your religion, starts from your education, your background, and your family trait. So as a result of that, in social sciences, we hardly employ only one theory. But critically looking into all these 3 theories, they speak to the same thing. Because the green theory has to do with sustainable development. It has to do with clean energy. It has to do with food sovereignty. The same thing is applicable to agroecological theory. But the only one that is a little bit different is ecofeminism. Like we are all familiar with, most especially students of social sciences. Feminism, we have different types of feminism. We have radical one, we have black feminism, African feminism, Western feminism. But ecofeminism has to do with food production, and it has to do with the role of women in the area of food production. And when we look into women, the way God creates women is unlike the way women are created, because women would like to take care of their own front. For instance, women can wake up one day and tell our spouse that, you know what, I'm moving to Kampala because I'm looking for a job. Or I'm moving to Pretoria or Johannesburg or Cape Town. Or I'm going to Abuja because my brother promised me a job. But for women, they will think of so many variables before they can move. They are not as mobile as you and I. For instance, in the Niger Delta region, when amnesty was introduced to those people who are causing problems in the Niger Delta, it was only men's show, forgetting that women are the only one that are always at the receiving end. And when it comes to food production, it is the women that are not stingy about their seeds and seedling. They like to share it with their neighbors. But for women, we're a little bit stingy, naturally. So that is why I'm going to call for or advocate for empowerment of women in the area of food production. Then, in order to empower women, we know that the wherewithal to promote food sovereignty is not readily available because of the usual demand of the collateral security. Women, because of the nature of Africa, whenever they get married, most especially in some tribes, immediately you get married, there is nothing, you don't have any business with your parents' property again. So, and when you get to marry somewhere, they are going to tell you that you are a woman, you are married here, you don't have any role to play. So as a result of that, many of them that are into food production, they embark on small scale in some cases, medium scale food production, of which they don't have collateral. And as a result of that, they need the input of birds in order to promote what they are producing. How they need birds in their production activities, I'm going to explain. A need for food security or food sovereignty from the global perspective. Come 2050, they were of the view that the population at the global level is going to be 10 billion. How are we going to feed the 10 billion people? We mustn't forget the politics of humanitarian assistance from the Western power. It's just like IMF and World Bank conditions, that as long as you are going to get this from us, this is what you are going to do. And that is one of the reasons why we failed to get it right in Africa until today. Because I remember when we were in university in 1985, by the time they introduced structural adjustment, structural adjustment program, so many conditions were attached to it. You have to introduce multi-party system. You have to embark on this issue of area of specialization, production of input for the manufacturing areas. At the end of the day, what we are producing before are the things that we are importing now. Now, Amatia was of the view that in order for us to produce what we need, we have to consider some variables: the affordability, availability, accessibility, and utility. For the purpose of this, I'm going to focus more on utility. But as a solution, we know the price of fertilizer now. Though we have Dangote refinery industry, which promised to produce urea fertilizer in Lagos, Nigeria. But is it going to be accessible to some of our people, considering the level of poverty in our midst? And there is a multinational chemical company called Yara. Yara receives nothing less than 10 billion US dollars every year as profit in the selling of fertilizer, but at what cost? So many farmers, because of the problem associated with climate change, whereby you cannot predict the availability of rainfall, it has taken so many farmers to bankruptcy. So now, what is the role of bat in this? Guano that we get from bat is a veritable source of fertilizer for what we are producing, most especially those people who embark on backyard farming or vertical farming in their backyard, or those people who embark on small-scale farming. And when we look into the role of the small-scale farmers, we realize that what they are producing is enough for us. Those of us who are from Nigeria, when you go to Mile 12, you see what these small-scale farmers are producing, that they are taking to Lagos on a daily basis. I don't know whether the same thing is applicable in Kenya. But when you look into the large-scale farmers, what they produce is not meant for the immediate consumption. Rather, they produce in order to serve as feeds for animals, to serve as industrial input. So what I'm trying to say in essence is that in an effort to make sure that we have enough beds in order to get enough guano to be encouraged. Then moving away from there, we know there is a plant called macadamia in South Africa. For I believe Professor Paul should know where we call Venda in Limpopo region. The vendors are known for production of avocado and mango. But now with the attraction of foreign exchange, macadamia has been taking over the production of avocado and mango now. And without bets, macadamia may not be able to produce enough fruit that we want. It's not only that. Figs, mangoes, dates, and peaches, those are the plants that needs the activities of bats in order to pollinate all these plants. So, what is the way forward? We need to encourage women in order to embark on small-scale farming. By interpretation, they will be able to get the beauty of birds. Then the use of insecticide and herbicide that continue to subjugate our biodiversity and ecosystem will be drastically reduced if we keep our birds. In conclusion, food sovereignty can be achieved when we encourage birds in our society. I can see that the moderators are traumatizing me. Thank you very much. So in the course of question and answer, I'll be able to explain further.
Thank you very much, sir. He has already used up his time, so we are going to actually take only one question or a comment. Because he has already used up his time, everybody has 10 minutes. Just a question or a comment.
10 minutes, presentation, 5 minutes question.
Yeah, you've already used up the whole 15 minutes.
The time flew so fast. And I know when professors speak, the inspiration comes so well. Yes, let's have 2 comments. A question and a comment, please.
Okay.
Thank you, sir, for your presentation. I want to ask more about the ecofeminism. I think I like the concept of ecofeminism. That is the role of women in moving conservation forward, in the using bats, especially for food security and food. Is it sovereignty now?
Food sovereignty, yeah.
Yes. So how do we come in this in this thought that you present to us? How, because I know you wanted to talk about that. I'm interested in how we come in as women. And that aspect where you talked about, it is the small-scale farmers that usually meet the need, not the large-scale farmers. Meeting the need in terms of food, the food that we buy in the market is from the small holders, small farmer holders, not the large farmer holders. Because I don't get that very, very well.
Okay, thank you very much. Regarding the concept of ecofeminism, like I indicated, we have different products in the theory of feminism. But for the purpose of this presentation, I focus on ecofeminism. When will ecology, feminism, women? What that means is that when you look into our society, most especially when you go to the deep rural areas, you realize that women are the ones who are producing food that we are consuming. Many men, they focus on cash crop farming. When I'm talking about cash crop farming, I'm not talking only about kola nut, palm oil, cocoa, coffee, zizal. I'm not only talking about that, but in some cases, maize, mealie meal, or corn can be equally be for human consumption, and it can be a cash crop, because men would like to sell whatever they produce. And if women are not careful, they may not even have access to what their husband produce. But it is women that will go and plant vegetable, plant onion, plant yam. This thing is very, very germane when you go to the Niger Delta in Nigeria, whereby men, you will see them tie wrapper in the morning, drinking. While women are the one who go to the Greeks, to produce, many official women who are producing in order to feed the family and to send their children to school. So what this theory is trying to say is that we need to encourage women, because women are very, very mobile. We don't look back anytime we want to take our decision. And from African perspective, we believe that we are the head of the family. So as a result of that, whatever we say is the final. Now look into the citing of basic infrastructure amenities. When it comes to borehole or anything, maybe health center, now you see men. Whereas the women that are bearing the brunt of going to the farm to get firewood, they are the ones who are going to the river in order to fetch water. Those ones may not be considered before they cite any social amenities in their community. So that is why we are calling for ecofeminism, so that at the end of the day, women are going to be recognized as a power to consider when it comes to food production, consumption, and distribution.
Thank you very much, sir. So, the next question, sir.
Yes. Okay.
Thank you so much, Prof.
You're welcome.
You are really opening new frontiers in ecofeminism. But then I wanted to, it's still wrapped up in my head. It's still foggy. I want to say, please, could you please establish the connection between ecofeminism and bats? And then you also talked about the role of bats in food security and food sovereignty. How are they able to establish that? I know you had so much to say, but for time constraints, but I would just like to quickly, so I will have a full grasp of that. Thank you, sir.
So, Prof, let me add this so that you can answer together.
Okay.
Yes, sir. When we're talking about let me use the word gospel about bat conservation.
Yeah.
Now, you've talked about ecofeminism.
Yeah.
Now, your advocacy on bats conservation, would you be focused on men or on women? Because you said men are so mobile.
Yeah.
And we don't know, I don't have the data or the information, whether those who actually try to take care of bats, you understand, by eliminating them, maybe it's skewed towards male or female. So your advocacy, because you're talking about ecofeminism, will it be towards men or towards women?
Now, thank you very much. I think that is the kernel of your question, which is being reframed. The connection between ecofeminism, food sovereignty, and birds is that the byproduct, the guano from birds, if we are able to get it in large quantity, it will serve as fertilizer for women who are in the deep rural areas, whereby they can use it to fertilize their plant. That is one. Number two, we know that we have two major types of birds. There are some that feeds on plants. There's another one that feeds on insects. Those ones that are feeding on insects will eliminate some insects. Statistics shows that in America saves more than $5 billion every year because of the bats that are controlling insects in their farms. So what I'm saying in essence is that using insecticide, it has so many disadvantages. Many people are getting terminal disease out of it. We are talking about cancer and so many other ones. For instance, about 3 or 4 years ago, Monsanto paid several millions of US dollars to some farmers who are using glyphosate in order to eliminate weeds in their farm. Then we know bats is just like a bee in terms of pollination. If we eliminate bees, there are so many plants that may not produce what is expected to produce, and we know the implication of that. So, in essence, it is an agent of balanced biodiversity. It is an agent of a very functional ecosystem. And we are talking about climate change. These birds, they pollinate, they drop so many seeds around. And these seeds, they will germinate, grow, and when they grow, they take in carbon dioxide and breathe out oxygen. So as a result of that, it's going to serve to some degree a balanced, clean environment. That is those are the. Your question? No, the link is that when you look into women, we are destructive naturally. But women, they think twice before they eliminate any animal, even plants. It is women that you find in extractive industry. When you go to Ife-Ijesa axis, you see how they destroy the vegetation, the forest there. And by implication, if we have some birds around that place, it's either they migrate to somewhere else or they are eliminated. So what I'm saying in essence is that men, they tend to use more insecticide and herbicide more than women, because women are the agent of sustainable development. That is my area of departure.
Thank you very much, Prof. Sir, for the wonderful presentation. So, because of our time, we are going to go to the next presenter. We will be talking on the might and misconception on Eidolon airborne bat in the environment. I'm going to call on Mr. Omotayo. Where is he? Let's.
So good afternoon, my distinguished audience. This afternoon, I'm presenting on behalf of the research group of Songoyomi, Ayontiji, Omotayo, and Jazat. And what we'll be discussing this afternoon is myths and misconceptions on Eidolon airborne bats in Iwo environs, Osun State, Nigeria. Now, a myth is a traditional belief or custom of a place that usually has strong roots and drives people's behavior to a very large extent. While misconceptions generally can be considered as incorrect or false ideas, howbeit people carry and run with it, and take it as truth. Now, with regards to Eidolon airborne bats, which are more common, the straw-colored but which are more common in this environment. I'm sorry, I'm trying to get a hand with the pointer. Can I have, okay, thank you. Okay, all right. Yes, we all are aware that bats are nocturnal mammals that occur ubiquitously in many forested regions of Nigeria. Bowen University campus is blessed to have quite a bit of tree vegetation since on the campus grounds, and so Bowen University campus therefore acts as a natural habitat for this particular bat species, particularly at the old campus of the institution. Now, bats have been long associated with human communities, and their flesh provides a ready delicacy for those who consume them. But we have the consistent issue of the negative perceptions that people still have in such in such a community. Even though this is an academic community, we still find that people still carry a lot of wrong perceptions as to, you know, what bats really do, and that may be quite surprising. So the essence of this study, we wanted to be able to identify some of the myths that, even though this is supposed to be an informed environment, as it were, what are some of the myths that people still associate with the presence of bats on this campus? And we wanted to also understand the level of people's awareness in terms of the impact of bats on the environment. We also wanted to have an evaluation of people's perception of the damage caused by bats in this environment, and to evaluate what are their responses in terms of the control of bat populations. So we sampled staff members for this study, and they were the main respondents, and we used purposive sampling method. We actually sent out 150 questionnaires, but we had 135 respondents, and then their responses were subjected to descriptive statistics. Now, in terms of the myths that we had, we came up with a number of myths, and we discovered that the myths that bats are blind were the highest. We had about over 50% respondents still perceiving that bats are blind, and that's quite surprising. Okay, so also we had a perception that bats are carriers of dangerous diseases, as a very high 59% actually of the respondents felt that bats are carriers of dangerous diseases. In terms of consumption, majority of our respondents do not consume bats, but about 11.9% of them do consume bats. We had an interesting statistic here when we asked whether bats were demonic, and we had 20% of the respondents having a strong feeling that bats were actually demonic. In terms of their benefits to agriculture, about 58% of the respondents were aware, or they recognized the fact that bats are good dispersal of seed, among other benefits that are listed, which include pest control, pollination, and pollination. Then in terms of the damage caused by bats, especially because Edelum helvum, the straw-colored bat, is a bat that tends to be more present among or close to human communities, we were able to discover from this study that the waste, that is the guano of the bats, were the main issues or the main reasons why people actually considered bats to be a nuisance in the environment. So we see that quite a high percentage of our respondents felt that bat waste and the stains that they cause were the major damage or nuisance that they felt bats' presence in the environment caused, aside other reasons. And then in terms of bat control, we came up with a number of parameters to ask what they felt would be the most appropriate way to consume them, to control them rather. And thankfully, about 65% of them felt that calling a professional bat control company would be the best way to manage their population. But it was quite surprising to us that 40% of the respondents felt that we needed to cut down trees in order to get rid of the bats. That's thinking of solving a problem by creating another problem. So, and then, of course, perhaps this stemmed out of the perception that majority of them felt, about 76% of them felt, that actually what was attracting the bats to this community were the plants and the flowers that are found in this environment. So, bats' presence on Bowen University campus actually is considered a nuisance to many of the respondents, or those that are the residents of this community. And that's because part of what they do in the course of their roosting activities are that they defoliate trees, which are significant components of a green environment. However, people are aware that bats do play important biological roles. They are aware of the fact that bats play important biological roles. However, because of the perceived negative, impact on the environment, they felt it was better to get rid of them all the same. So these conclusions, as it were, made them feel that calling for an adequate professional management of the bat species within the premises would be the best way to control the bats. In conclusion, we're able to discover from this study, although rudimentary as it were, that bat roles in the environment are affected by the perceptions of the people who interact with this species. And we discover that the people in this environment are well aware of the positive impact of the bats in the environment, with the highest perception being that they are good pollinators. However, they also perceive that the stains caused by the guano or the waste of the bats were the most negative impact that the bats have on the environment, aside the noise and other and the odor that they produce as well. And they felt that the engagement of professional service would be the best way to control the occurrence of bats in the environment. These are some of the references we considered, and thank you very much for your attention.
Thank you for the wonderful presentation, sir. So I don't know, maybe anyone has any question or comments. Okay, sorry. Okay, sir. Sorry.
Okay, thank you. It was, it's actually an eye-opening presentation. But your conclusion and your results, they differ a little because you actually focused on members of staff.
Yes.
In Bowen University.
Yes.
So it will be difficult to extrapolate it to the Iwo community.
Yes.
Even within Bowen University, there are students, you understand? Now, I don't know if you categorized the members of staff, probably based on senior, junior, you understand? That is one thing.
Okay, sir.
It may look like a homogeneous group. But with the first presentation about ecofeminism, you may also want to put gender. Let's look at how the males, you understand, see all these perceptions compared to the females. These are just some additions that you can...
Thank you very much, and noted. Actually, we consulted a few students also responded to this. Okay, so thank you very much for the observation.
Thank you, Doctor, for that presentation. Just in line with what Prof just asked, I want to inquire whether you actually you add some other peculiarities for your respondents than being members of staff or students of the institution, because otherwise those respondents, if they are not categorized based on certain things out of which we are now making our conclusion now, it may be somehow shaky.
Okay.
For instance, you saw that some respondents said that bats defoliate trees. That one can be visibly seen. But for those ones that are saying that bats are friendly to the environment, what are the basis? Some say they are demonic. What is the basis? Have they experienced such for that kind of conclusion? Thank you, sir.
Thank you very much.
Thank you for your presentation.
Thank you,
I just had a question about when you had a slide on the misconceptions.
Yes.
You put something there about disease. And I wonder if that can accurately be called a misconception, or there is an element of truth to it.
Okay, thank you. Yes, okay. So actually, it's a myth that bats are carriers of dangerous diseases. We have research to establish that bats do act as carriers of zoonotic and also vector-carrying organisms, as it were. But the idea that bats the presence of bats means, and Professor Webella spoke to it when he made his discussion too, the presence of bats immediately causes disease to come into the environment is a myth. We can call that a myth. So, and that is why we included it as one of the myths that we felt should be noted. So I don't know, it's like Prof, you're raising it. So.
But that another question that has been raised in many fora. The fact that you find antibodies, even in you, doesn't mean that you are a disease carrier. So often, many people, and because it's a goose that lays the golden eggs, many people are going out killing bats looking for disease variants, and they find antibodies. And that is equal to, wrongly so, that those bats are carriers of disease. Not necessarily. They are also fighting the disease.
Thank you very much, Prof.
Thank you for the wonderful presentation, sir. Let's give him a round of applause as he goes back to his seats. Okay, I'm going to call on the next presenter, which is Mr. Guam Thomas, a checklist of ectoparasites of bats, diversity and implications. Thank you very much, sir.
Slide presentation, please. Just the one down. All right, thank you. All right, good afternoon. I stand on the existing protocols, and I'm here by name Guam Thomas to present the research finding on the checklist of ectoparasites of bats, their diversity and possible implications. Below are the list of the co-authors of this research. We all know that bats are mammals, and largely we can classify bats into two major groups. The ones that feed on fruits, we call them the frugivorous bat, and the ones that feed on insects, the insectivorous bat. In that class, you find the other species that lap on blood. So basically, with that general knowledge of bats, we've been trapping bats to identify them to species level and to check their relationship with other animals like insects and arthropods in general. So we know that ectoparasites play a significant role. Most arthropods are of medical importance. They play that role of transmitting diseases. We've seen the case of mosquito carrying malaria and all that. So it's with that knowledge that we want to check what's going on with the bats. So ectoparasites have diverse way of evolving along lineages. So we want to check if that also is the case with some bat species. We know that some parasites can be obligate in some particular species of animals. And that means those parasites tend to spend a large chunk of their life on the host. If it's a bat, then it's solidly found on that bat. The parasite can't live off that particular bat species. That means the biology of the parasite is tied to the biology of the bat, meaning reproduction, feeding, and all that the parasite will need will have to be on the bat species. Therefore, that may impact the fitness of the bat itself. This study aims to compile a checklist of all the parasites that associate with the bat species that we captured. So we focus on the diversity and the distribution and the potential health implications of those species. A method was by sampling the bats themselves using one of the methods, which is the mist netting. We identified the individuals we captured right from 2001 to 2003. Some of the bat species, which were 10 in all, some of them don't have the ectoparasite on them. So that was a negative for the ectoparasite presence. Then for those that were positive for ectoparasite present on them, we carefully check the body and we isolate the ectoparasite from them and carefully take it into the laboratory for further identification and taxonomic classification. So we store them in ethanol, refrigerate them till the time that we want to do the identification, and we use that microscope there in the laboratory to check and know whether it's an insect or an arthropod, and identify them to their particular. We stop at family level because identification for insect is quite technical, and for bat flies particularly, it's just an emerging field, right? The result we saw from the 10 species of bat we identified were the bat flies and mites and an arachnid. That's a picture of one of the arachnids there. Okay, the result shows that most of the, Rosetus aegyptiacus, which is one of the bat species, tend to carry more by number now, the ectoparasite compared to the others that are there. So checking the groups of bats, we find out that the frugivorous bats harbor more of the ectoparasite than the insectivorous bats. Possibly the insectivorous bats may end up picking the ectoparasite from them and eating them. They're insectivorous bats, they eat insects. So that shows us that more of the ectoparasites were on the frugivorous. Then we also checked, the ectoparasites families now. After identifying the ectoparasite themselves, not the bats, the ectoparasite, we still found out that they were more on the frugivorous, more families on the frugivorous than on the insectivorous. Then the bat species and comparing the count to the groups or the families of the ectoparasites now, the Nectibidae happen to be the largest family, which are the bat flies now. Then followed by the bat fleas, and 20% was recorded as the Sphinxinidae, which are the arachnids. And basically, some of the species are overlapping some species, and some, for example, the Nyctiribidae is only found in Rosetus. So that we draw, we notice that the Nyctiribidae is mainly a Rosetus case, which suggests possibly is an obligate of that particular bat species. And though it's found also on Tadarida species, then Ixnopsilidae and Sphinxtonicidae were parasitizing species like Scotophilus, and only on that particular bat species. So in conclusion, this study has given us a glimpse to the ectoparasites that are on bats, and some that are obligates on some species, and the possible medical implications that we may have on this particular ectoparasite, because there have been reports on the ectoparasite harboring some pathogens like rhabdoviruses and the others. Thank you very much. Okay.
Thank you very much, sir, for the wonderful presentation. I don't know, maybe anyone has a question or comment. Okay, Professor.
I have a comment. Nyctiperidae, I think you need to do a lot of reading, because Nyctiperids are not obligate ectoparasites. They are found, and in your study it shows very clear they are found in Tadaridae, in the family, in the genus Tadarida. So they are not just on rosetas, they are also on Tadarida, so they are not obligate. But number two is that not that insectivorous bats eat them, no. The reason is that your bat studies that were sampled are not colonial. Okay? They are not colonial roosting. Rosetas is colonial roosting. So bats that are found roosting together in a colony have more ectoparasites than those that roost like Tadarida. Tadarida roost 1 or 2 individuals. Okay?
They are colony nesters. So
Yeah. So, no, they are not many. They are not as many as rosetas. So if you look at some of the species that you found, they either roost few individuals or singly, not many. So the reason is that ectoparasites are found because they move between individuals. In the colonially roosting species like Miniopteras, like horseshoe bats that roost many, like leaf-nosed bats, you find those ectos in those species that roost in colonies. But if they are roosting alone or 2 individuals like Epimetheus,
yes,
you don't find ectos. Okay, so it's not that they are eating them, it is that they are roosting either as individuals or small individuals of 10, 15, 20. Okay. So that's the reason.
Yeah. Thank you, sir. That comment.
Thank you for the beautiful presentation. My question, on the bats you collected, did you get different species of bat flies? Or is the same kind of bat fly you got on a particular bat?
Okay, we class on a particular bat, mostly is one species we got. But there are few cases that we got 2 species of the ectoparasite on them.
So on some of the bats, you had different group of arthropods on a particular bat?
Yes.
Okay.
But mostly it's not more than 2.
Okay.
Yes. Sir? All right, sir. Yeah, right.
Thank you. I think to add to this particular study, not just getting the ectoparasites, we are in collaboration with other scientists who are quite interested in these ectoparasites to look at them again and check whether they carry certain viruses and all that. And I think that's something that will come much later. Because when we had this one at the presentation, quite a lot of people are interested in the ticks, and they should be carrying something. Thank you very much.
Okay, I think from a bat study, we also made a publication just last month. It's on ectoparasites. And just like our presenter has said, we could only get one arthropod, a Nycteribidae, from Idolon. Although there's a publication that says they could get a Nycteribidae and another arthropod.
Yes.
There's a publication that says that. But while reviewing, I think some of the reviewers commented that I should be very careful with the word, since we did not actually sacrifice the animal to bring everything out of the four, that there may be some other organisms, mites that are smaller. So I was very careful in using some of my words. So it's possible that there may be some other mites, very tiny, that one may not be able to visualize. And the one that we saw, Cyclopodia griefi, you understand, is the one. And we also know that even this ectoparasite also carries another parasite.
Yes, yes.
Thank you, sir.
Right. Thank you. Thank you very much.
Thank you for the wonderful presentation. Can we give him a round of applause as he's going back to his seat? So I'm going to call on the next presenter, which is Mr. Stephen Ezekiel, talking on the topic coronavirus detection in insectivorous bats, a study in Yankari Game Reserve.
Okay. Okay, good afternoon, everyone. So I'll be presenting on coronavirus detection in insectivorous bats at the Yankari Game Reserve. So my name is Stephen Mayo Ezekiel, and these are my co-authors. Okay. So the outbreak of COVID in 2019 necessitates this kind of research to actually see the sources of coronaviruses. And just like the professor said, that bats had actually been actually accused to be the source of coronavirus, but we can't generally say what are the sources of coronavirus. Hence, to understand the prevalence and the diversity of coronavirus is essential in public health information and wildlife conservation as well. So the aim of this work is in general to determine the prevalence of coronavirus in insectivorous bats at the Yankari Game Reserve. Now, Yankari Game Reserve is one of only the tourist sites we have in Nigeria, and there's an average of 10,000 tourists visiting the Yankari Game Reserve every year. So making this place a very important place to study and for conservation studies. So bats were trapped using mist nets. Morphometric measurements were taken of mass, sex, and body lengths. And for this study, 52 bat individuals were subjected for this analysis, and they are scattered around the banana pipistrellus, the Rhinolophus landeri, the Tadaridus species, and the Scotophilus species as well. Now, non-invasive methods were used in sample collection of oral swab and rectal swab using a swab stick, and this was transported for analysis in the laboratory using the viral transport medium, which is the VTM. RNA extraction was done, and amplification of coronavirus gene were done. Our primers were actually designed to amplify every gene of coronavirus in that sample. So we're just trying to explore the total prevalence, every possible coronavirus that we can find in that sample, if they are positive of coronavirus or not. And these are the count of the bat. Tadarida species seems to be the highest individual bat that was trapped in this study. Now, and out of the 52 samples that were subjected for this analysis, 46 of them tested positive of coronavirus, which is 88.46%, meaning for every 10 bats that is trapped, 8 is tested positive for one form of coronavirus or the other. This is showing high prevalence of coronavirus in the bat in Yankari Game Reserve. Now, the CT value was used to determine positivity of our samples, and CT value between 30 and 37 indicates moderate infection load. However, still tested positive for coronavirus. Now, the coronavirus in alpha and beta coronaviruses have been extracted from bat gene pool. So that leaves us with either HKU 4, 5, and 9, or 2 and 8 to actually give us a range of the possible coronaviruses that we could find in this population. However, this study is novel in Yankari Game Reserve, giving us a choice that there might be either novel viruses that are yet to be detected in the population. Now, why is this study very important? The ecology of these birds and the influx of tourists makes Yankari Game Reserve a possible place for virus spillover, and because the ecology of these birds suggests and that they roost around residential areas. And the background information, during our sampling, speaking to one of the local guide, he told us one of the men cleaning around the bat roost at a certain point developed a symptoms of hemorrhagic fever, which is spitting of blood, giving us an indication of viral infection. So Yankari Game Reserve remain a very good hotspot for virus spillover, hence, and there is need and to ensure the conservation of this bat and reduction of human-wildlife interaction in Yankari Game Reserve, and also in planning public health strategy in the cases of virus spillover. And also, and there's need for further studies and to detect the specific strain of coronaviruses in the population in Yankari Game Reserve. Thank you very much for listening, and I'd like to give credits to this institution. Thank you very much.
Yeah, thank you very much for a very wonderful presentation. So please go to your results section. So you had a very high prevalence of like 88.46%. And then you also showed us in your methodology that you used a pan-coronavirus assay, right? That targeted all the genes.
Yeah, sure.
Don't you think there would be room here for false positive? How did you check? I think the question is, how did you ensure that what you got here were not false positive?
Okay, so
because the prevalence is actually quite very high.
Yes, yes. Yeah, that was surprising as well. And because this is a novel study, like this is the first time we're actually engaging in this kind of investigation. But our CT value, which is and the number that's the threshold circle, right? It gives an indication of the viral load. However, our CT value indicates low parasitic load, right? Because it takes 34, between 30 to 37, 38 cycle, and for the genes to be amplified, which indicates that even though the viral load are small, but they indeed indicate that they are positive.
Okay, so I work on coronaviruses, okay? And I've published extensively on coronaviruses. Yeah. That's why I asked that. I would rather have was it a positive control to like show, and then did you have like a G block or something to show the concentration? And I think that would have helped modify whatever finding you got. And secondly, I would have preferred, there are some genes that are specific or conserved coronaviruses, which you can use to be sure that you can type properly. One of them is the RDRP, which is the main assay used for coronavirus typing. And then there's some WHO protocol that use ORF1AB, the N gene, the E gene. So it gives you a higher level of reliability of what you're getting. I would suggest here you start first with a conventional PCR assay, a pan-coronavirus conventional PCR assay. And then go downstream to address that. I have used this before. What you're getting here is false positive. From experience. So if you use that with a control, that would give you a higher reliability of whatever thing you're finding here.
Thank you very much.
The prevalence is quite very high.
It will be that prevalence is so high against any conventional methods. So I'm not a ecologist like he is, but prevalence rate of that of viruses is highly, highly doubtful, and it will be totally rejected at the territorial level.
Thank you very much, sir.
Is going to just maybe in a week or so, and I will discuss further. But the aspect of this is where we look at it at a One Health program issue. That's the level that we are coming. We are the West African Center for Emerging Infectious Diseases. We are looking at it at that level. So when we have this, our other teams, the vet team, the clinical team, the mosquito team also came in. So these are all work in progress. They came in to take samples so that we'll be able to say at the end of the day what actually is happening. So it's a One Health approach. This was just an aspect of it that we just took out. Because when we saw that, we were also disturbed. More so, that's a tourist area. So there are so many things that might be happening. So we are moving forward about that. Thank you.
Thank you, Mr. Stephen, for the wonderful presentation, sir. Let's give him a round of applause as he goes back to his seat. I'm going to call on the next presenter, which is Dr. Taye Adeyanju, together with Mr. Okpanachi Monday, talking on rapid survey of bat genetic diversity in selected sites in Nigeria. And for the last presenter, I think you should be, that's Miss Ayokunle Oluwadamilare. Please, get ready.
Okay. Okay, good evening, everyone. I'm Adeyanju, and of course,
I am Okpanachi Ikwojo Monday.
Okay, so we're here to present presently on rapid survey of bat genetic diversity in selected sites in Nigeria. Well, we all know that bats in general are not uniformly distributed along land uses. If we look around Bowen, many of you, of course, will agree with me that you do not feel that there are lots of bats around Bowen. But of course, we do have different land use types in Bowen. We have some places that are heavily vegetated. And of course, the probability is, of course, that in many of these areas, we will have more chances of finding higher degree number of bat species. And it's very also important that bats are not only morphologically identified. We also have bats being identified through acoustic means, for example. And of course, bats being identified also through molecular material, that's DNA. And in coming to identify bats, we know that there are a lot of misidentifications that come with bats. When we started, it's not on. When we started bat work as far back as 2010, we discovered that there were lots of areas where we thought we had some species, and at the end of the day, we found out that those species did not actually even occur in the entirety of Africa. So there's a lot of anomalies that come around when we depend on just morphological parameters to identify bats in the field. Also, we know that in identifying bats, there are lots of times we have to go into brain case measurements even to be able to distinguish between sister species of some peculiar species like Pipistrellus and so on. So in identifying cryptic species, we decided to use two techniques in this area, the morphometrics, which we've been using all along, and then combine that with genetic material, DNA. And we've been able to bring out a lot of new knowledge that is going to help us in this ongoing project of the WAC EID. Of course, morphological identification involves taking forearm measurements, tail length for insectivorous bats, weight, and so on, wing lengths, and then comparing that with keys. For example, the bats of Nigeria, if we're working in Nigeria, or then bats of Africa. And these are the various anatomical features that we have for the bats. Of course, we have the wings. For those of you who have not held the bats before, if you look at the tail region, only the insectivorous bats, of course, have that, while the fruit bats have more of a false tail. So it's possible for us to measure all the way from somewhere around the anus there, all the way to the tip, and that's taken as the tail. Then the insectivorous bats also have a tragus, and that's it from there to there. And then the ear length also from there all the way to the breadth there. So, and then the wing length is also taken, though it's not a standard measurement but when it comes to bringing as many other variables, wing length can also be taken. But one of the main other things that is measured is the forearm length, which is from here, the tip, all the way to there. So there are a lot of diverse bats that we come around in the field, and these are bats that we trapped in Nigeria during the WAC EID project all the way from 2021 to current. The project is still ongoing, but of course we've had several very lovely species. Among them, very, very easy to identify bats like these fruit bats or dog-faced bats, very easy to identify. But then we have very tricky bats like the banana pipistrellus, which of course we have lots of species within a single genus. And then we come into the conclusion of just giving them Pipistrellus SPP and not being able to know exactly what species they are. And that's the reason why, of course, we go molecular. So Monday will continue.
Thank you very much. So during the course of this work, bats were captured using mist nets, and DNA was extracted from their wing membrane biopsy. So after collecting the wing membrane, that is the wing we amplify the mitochondrial COI gene using the following set of primers. Okay. So using the following set of primers, we sequence after amplification. So after we sequence, generated mitochondrial sequences were checked for ambiguous peaks, and we trimmed to eliminate the primer nucleotide sequences. So we confirm, we kind of do a correlation between our phenotypic identification and our genotypic identification after we have run our generated sequences through the NCBI BLAST database. So then after that, we use genus software to construct a maximum likelihood phylogenetic tree using the best fitting model based on the AIC, which is the Archaic Information Criterion. So this is the corresponding genotypic identification in respect to our own phenotypic identification using the guide. So we can see that banana pipistrellus identification on the field was mostly misidentified for Ipposideros ruba, Neuromycia pipistrellus, but if you see for some species like Epomophorus gambianus and Ipposideros and Megaloglossus, Rhinolophus, Rosetus, and Scotophilus were mostly identified correctly. Tadarida species was identified because of the how will I put it now? The taxonomy shift in the current taxonomy updates in bats identification. Tadaridana is mostly identified as cheiroforms. So that's why it is like that. So next, we try to check for the sensitivity, specificity, and positive predicting value, negative predictive value of the phenotypic identification for with respect to different bat species. So the sensitivity helps us to know the proportion of the true positives, while the specificity helps us to know the proportion of the true negative. The positive predicting value and the negative predicting value help us to know the how a chiropterist identifies this various species based on the true positive and true negative, respectively. So we see that species like Scotophilus, Epomophorus gambianus, Micropterus, Rhinolophus, Rosetus, and Cheiroform species all have perfect sensitivity, specificity, PPV, and MPV scores. But we see species like the Glauconocteris, which had 14.29 sensitivity and a positive predicting value of 14.29, means that to identify this species it means that it has a very low, the chances of getting a positive result, like identifying it, is quite low due to the morphological relationship, and because of the morphology, you cannot like differentiate it morphologically. It's not morphologically distinct from other species, so that's why it is like that. But you can see that the specificity is quite high, which means that you can actually say this is not Glauconocteris, but what is it? That is true negatives. That's what the specificity does. It helps us to identify the true negatives. So now for the phylogenetic tree, well, this phylogenetic tree tells us a lot about the genetic diversity of this bat. We can see here, this particular clade here, that the Micropterus possilus and the Epomophorus gambianus form a single clade. What is this telling us? This means that, as we all know that the mitochondria, the mitochondria DNA, which we have for things like the COI, cytochrome B, cannot differentiate, distinctively tell us the difference between the Micropterus possilus and the Epomophorus gambianus, which means probably they are paralogs. But if you look at this species distinctively, physically, you know that Micropterus possilus is very different from Epomophorus gambianus. And this particular result is also backed up by another study which is done somewhere in Africa too, which states that these two species cannot be differentiated using mitochondrial DNA, but you have to use other part of their genome, which is the nucleate DNA. So also we see that for Eposideros, we say that there is the Eposideros pomona, they have two different species that shows two different preference to two different sites. The Eposideros pomona in Indonre is different from the one in Yankari. The one in Indonre is the one in Pomona, while Eposideros roba is the one in Yankari. We can see that the cheiroforms are genetically diverse. You can see how they form different, their class and their branch lengths are quite different. What is this telling us? It means that this particular species, not just cheiroform, even Neuromycia and Pipistrellus, they show genetic diversity. It means they are adapting to the current changes in the evolution with response to the climate change and probably human activity disturbing their ecosystem. So discussion, this result shows us that some species morphological identification correlates with a genotypic identification, such as Epomophorus gambianus and Rosetus aegyptiacus. Some bat species add more than one possible genotypic identity. What is this saying? It means that even for genetic identification, bat species are very quite closely related, that it takes more than just one gene to distinctively tell you the difference between a particular species, especially if you want to identify them, to to the species level. The result shows the phenotypic identification can accurately identify specific species. Nevertheless, genotypic identification should be used for cryptic species and hybrids. Phylogenetic analysis shows Epomphoros gambianus and Micropterus bacillus shows no distinctive speciation despite being morphologically distinct. As I said earlier, Scotophilus pipistrellus caeriform shows genetic diversity. Eposiderus promona and Eposiderus ruber show preference to two distinct ecological zones in Dore and Yankari respectively. Molecular characterization is an important tool for the conformation of cryptic species such as cryptic species as bat, while for distinct morphological groups, morphometric keys are sufficient for identification. Conclusion: the study presented a degree of correlation between phenotypic and genotypic identification of bat in Nigeria. Findings from this study shows the significance of the genotypic characterization for the correct identification of some cryptic species of bat. Inferring the phenotypic identification can be significantly inaccurate. For distinct species, morphometric keys could be employed for identification, such as species like Rosetus aegyptiacus, Scotophilus, Epomphoros gambianus. Banana pipistrellus and Tadarida shows in multiple phenotypic identification, which means that this particular species can be easily misidentified phenotypically. I acknowledge the following institution. Thank you.
Thank you for the wonderful presentation, sir. I don't know, maybe anybody has a comment or question. I'm going to take just one comment or question. Okay, just one.
Yeah, thank you very much. You guys have done a really, really great work. But I just have a couple of questions. The first one is, was there like a threshold for, or like a species demarcation criteria using the genomic sequences? Was there like a criteria at what percentage of your blast search would you say, oh, it's related to this, right? I think that's one. And then two, okay, you only use the cytochrome oxidase one gene, right? And not the cytochrome B, because what you're experiencing, I had the same, I think I had a similar experience too, but I also did cytochrome B along with cytochrome oxidase 1 and compare. I think the results were different. But I'm just particular with the criteria. So like your blast search, what percentage of the match did you conclude that, oh, it gave you this?
Yeah, okay.
I think what you are doing is just part of the puzzle, because you're looking at cytochrome B, or you're looking at only one part of the gene.
Yeah.
And so what we are doing in taxonomy is we're looking at what is known as integrative taxonomy. You look at not just genetics, not just morphology, but also you go further than that. You also go further, and you guys were not even going to the gene bank to look at type specimens, to look at original specimens. So you're not comparing with that. So what we are doing here is just part of the puzzle. We also look at chromosomes. Okay? We also look at, for insectivorous bats, we're also looking at sounds. So that for cryptic species, you can't just rely on molecular. Molecular analysis is not a magic wand. You go beyond that. You also go look at other aspects of the nuclear, it's called nucleus. Not just the mitochondrial B, but also you also go into the nucleus of the genes. So you look at all aspects for you to be able to limit species that are related to our sibling species. And what you're looking here is just one part of the jigsaw puzzle.
Thank you very much, sir. For your question, sir, you asked about the criteria that I used before I could say, okay, this particular species belongs to, this particular sample belongs to this particular species. Well, when blasting, the percentage identity was around 100 to 99 for many of these sequences. And also we use, I used also the e-value, but also too, I would like to say that while blasting, many of the sequences came positive for probably, because even as I look at the first 10 sequences and I check, okay, are they correlating? If they correlate, it gives me probably a high confidence that what I'm doing is correct. And also, once I do it, I also I check it with the phenotypic identification if there's a rhythm there's a trend that is going on. But nevertheless, some species don't tend to follow that same trend. Like, let's say, for example, the banana pipistrellus. It is quite difficult, even for the large sequence, you will see a lot of overlappings between that particular species and other species too. So yeah, I think it's something that I would like to take probably the advice that Prof have said, because genetics alone doesn't really distinct bats, which is quite different from other species of animals. So apart from genetics, you can also use probably the acoustics, which is something that I'm trying to also venture into now. And also probably because of we're a conservation institute, we can't really say want to sacrifice the animal to check for their morphology and their skeletal, and also. Okay, sir.
So, sacrificing specimens is critical.
Yes.
You can't talk about morphology unless you're looking at the teeth, unless you're looking at the skulls. What you are measuring is just external measurements.
Yeah.
But unless you sacrifice and look at the craniodental measurements, then what you're talking about is just superficial.
Okay.
Because you also have to take measurements of the skull and teeth, so many aspects of the skull.
Okay.
And that specimens, in cases where you have to describe a new species, you can't describe a new species without a specimen.
Okay.
So collecting specimens are crucial.
I think we agree with the collection of cranial measurements, and we've actually started doing something with that at Ibadan. And I think we've not done that on a large scale, but of course we're beginning to do that. I think the issue actually has been we're definitely going to have to do some more collaborations with museums outside of the country for us to get to do that. And I think because of the lockdown and everything, we couldn't do that, but we're coming there.
Yeah, sorry, just to also.
Yes, because. Yes. Yes, we agree with taxonomy. In fact, on the gene bank, many of the names, we got mixtures of the names, and of course that's where some of the confusion, because of course we need to, when a geneticist is looking at ecological data, sometimes he just analyzes it basically on genetics, and then, but we could give them some more eyes to see exactly why there are some differences. And of course, one of those reasons is taxonomy. Another reason could also be due to contaminations during collections. So we agree with that.
Yeah, sorry, just to chip in also, Gen Bank is sometimes a little bit skewed, okay? It might not give you the true picture of what you truly want, especially with regards to sequences from Africa, virus, bacteria, and including bats. Very few sequences from Africa have actually been deposited in Gen Bank, and very little in terms of genomic studies have been done. Those are some of the challenges. For me, this is a question that bat researchers should take home. This same question was asked for the Human Genome Project, because we kept using morphology to identify. Honestly, the solution will still lie in genome, because the genome of every organism has a kind of message hidden there that will help you understand that particular organism. Until we can do a full genome of some of the bat species, that will give us a true picture of what we're actually dealing with. That has not really been done in Africa. I think that should stimulate our minds that the next level should be, can we start having full genome of this unique bat species in Africa? just like the Human 1000 Genome Project, right? Today we can clearly say, distinguish different humans, right? The Caucasians, the African, and all of that, based on their genome. And then precision medicine is also going to be based on genome. So understanding the genome of bat is really, really key for proper identification of the bat. The morphology is key, very, very important. But every message of every organism lies in their genome. What we are doing is just a surface until we start having full genome sequences of this bat species. Until then, we will still just be running in cycles.
Thank you. I just wanted to make a comment when I spoke about sacrificing. You will appreciate our background. We come from a conservation biology institute, and I am the PI for this particular work. Before we started, you know how these Americans do. There are strict rules, protocols, and all that. And so we had ethical clearance before we started this work. So we never had the opportunity to actually slaughter. But in most of our discussions and some people who have found, the problem we had was these insectivorous birds having their blood. The blood was so small. And when we contacted the people in the lab, how are we, they said this blood is too small. Damn you, we can't do anything about this. We can't get the serum, we can't do that. And then again, we are seeing some of our counterparts in other places, they are doing, so how are you doing it? They said they were sacrificing the bats. I said, oh my God. We had a little opportunity in Gendry, you remembered, when some of the bats got dead, so we had to remove the organs and such. But now, we have to look for another clearance, another ethical clearance, so that we will be able to sacrifice the bats. I don't know if there is another way around it. I see Prof standing up.
You can justify and it's ethically approved to sacrifice a certain number of individuals per species for the sake of science, and that is allowable. So that can be, when you write a proposal, pass it through ethical animal ethics committees, they also approve sacrifice certain individuals. Yeah, so you have to do it, and they accept that because it is science.
I think the debate on this topic, because we trashed this issue with Professor Atobatele, with some of the other people with VET, is that if we have to open up the skull of every bat we catch in the field, then, because, for example, the Pipistrellus, completely impossible for us to get identifications directly on cue in the field. So, and we come in Alma Forest Reserve, we come across so many of these Pipistrellus, even in degraded forest areas. So what's the way out? I think that's the issue. So that, for example, we can say exactly when we catch a bat, maybe through the echolocation or through some particular measurement or through DNA, we can say exactly this is Pipistrellus nanus or this is Pipistrellus whatsoever. I think that's the challenge right now.
Thank you. I'm still a novelist in your area. But from all indications, from what I heard, as per the deficiencies in the work, which I think you have admitted, can be resolved with the title of your work. So I guess this is a rapid work. You said rapid survey, isn't it? Yes. So I think, it's pardonable. All those deficiencies, and you said in the course of your presentation that the work is still ongoing. So, well done, sir.
Thank you very much, sir.
Okay, thank you very much, Dr. Otayi Adeyanju and Mr. Monday Okpanachi for the wonderful presentation. Because of our time, I will go to the next presenter, which is Ms. Ayokunle Oluwadamilare, talking on isolation and molecular characterization of bacteria isolated from Edelon airborne droppings in Bowen University, Iwo, Nigeria.
Good afternoon once again. My name is Ayokunle Damilare, and I'm presenting isolation and molecular characterization of bacteria isolated from droppings of Idolon Evum in Bowen University. This is a collaborative work, and these are the team members. We have microbiologists and a zoologist, or a wildlife biologist. So talking about Idolon, we have two major species, and that's the Madagascar fruit bat and the straw-colored fruit bat. Of course, we worked on the straw-colored fruit bat, Idolon Evum. They are known to, bats generally are known to be reservoirs for mammalian pathogens, including virus, bacteria, and fungi. Now, in large fruit bat roosts, we discover that their dropping can accumulate over time, creating piles of their excrement, which is called guano, which can run several meters deep and form complex interaction of organisms in an habitat. And then as the guano decays over time, the nutrient composition changes and the bacterial community become deep within the pile. And majority of these have not been characterized yet because bat study is still upcoming, what we have so far. So we have the general major characteristics of bats, their ability for flight, their longevity, their noise, and they are nocturnal animals. We've already established a majority of all this today. The objective of the project was to map out roosting locations of the fruit bats we have on campus, and then to isolate bacteria from their guanos. Then to identify the bacteria isolated from these granules using the Gram reaction and biochemical tests, then molecular characterization. The study area is Bowen University, where we are right now. And majority of the bat roosts are on the other side of the campus. That's the temporary side, the old site. So that's where we got most of. So we have some identified bat roosts already, and they're by the right of the image we can see projected. Then, this is a very good example of one of the bat roosts we have in Bowen, and then the bat dropping. Actually, this is actually on a cemented floor. We have, directly under this street, take for instance, it's not cemented. So we have them all around under the roosts, some are up to piles and all of that. So that's where we got the bat droppings we worked with. And one of the objectives is to map out the bat roost locations which are projected. Then the fresh samples were collected overnight with the use of black polythene bags that were left underneath the tree roots of the selected sites at sunset at 6:30 p.m. till very early 6 a.m. in the next morning, where they are gotten and air dried. And after that, we're able to work with them. Then at the site of collection, the following morning, patches and guano pools were found, and then these are what we worked with. Then a total of 5 fresh guano pellets were collected from the bag, which is used for further studies. We were able to get from 5 roosts, mango tree, castor tree, and almond tree. After which there was experimental analysis, culture-dependent analysis, then the biochemical test. And we had our result before we took it for molecular characterization. And we had Staphylococcus bacteria identified. Then after the samples were taken for molecular characterization, we found Staphylococcus, then we had Bacillus species of bacteria also. And okay, here is the phylogenetic tree analysis of the samples that were gotten. And at the end of the day, we were able to validate that bat guano can be accessed for bacterial colony, which are of research, industrial, and commercial potential. Another interesting thing is that the bacterial species that were gotten are are not pathogenic. They are not known to cause disease in mammals or humans. So, and mostly collected were single bacterial group, which are gram-positive bacteria. Thank you very much for listening.
Thank you very much, Ma, for the wonderful presentation. I don't know if anybody has a question or comment. Okay, sir. Thank you, Ma. Use the mic, Ma.
Okay, thank you very much. Wonderful presentation. I think you just ran through those methodologies. You didn't really tell us how you got to isolate those pathogenic bacteria that you're talking about, or let's say beneficial in your sense of it. Okay. Okay. All right, thank you. I would have loved you to, when you were presenting, just talk about it, just run through it. So I was afraid that you got the three particular bacteria and there might be a false positive result. But, okay, that's okay.
Okay, Professor Woshini has a comment.
Good afternoon. Good afternoon, Ms.
Good afternoon, ma'am.
Yeah, thank you for the presentation. I just wanted to ask, your conclusion said that they were not pathogenic. Now, how did you confirm their non-pathogenicity?
Okay, they don't cause diseases.
How did you confirm? Okay, let me just let me just pass a comment, okay, and not stress us by asking questions. Okay. The fact that literature has shown us that maybe a particular species or genus are non-pathogenic does not mean that the actual one that is isolated from that environment, okay, might not be pathogenic. It's just like saying that if people from a particular race are nice, you can still see one or two that will deviate. It might be their pathogenicity could be means of survival. Okay. So for us to conclude, I think it will be a bit unscientific to conclude that they are non-pathogenic. If you have the isolates, we can test for pathogenicity just to help and to be sure that whether they are pathogenic or not.
Thank you so much, ma'am.
Thank you, sir.
All right, do we have any other comments or questions for her? All right, I know it's not easy to stand up here and have to be put in a hot seat, even when you know you have produced an intellectual product. I want us to gratefully celebrate everyone who stood here to present in the last few minutes. We really appreciate you. Thank you so much for your presentation. And at this point, I want us to also celebrate the man who anchored in the last few minutes. That's Mr. Oladeji, Israel. Let's put our hands together for him. He's not here as an anchor, he's here as a participant. And he got the job of anchoring, and he decided to do it like that's what he came for. We are grateful. Thank you very much. At this point, we have an equally important segment of this program, which we have waited for and look forward to. And that is going to be the bat genomics training and talk that we're going to have, and that's going to be done by a collectivity of people. Dr. Taiwo Crosby, Dr. Uwem George. Are they all here? I think one of them stepped out just now. Okay, he's here. All right. His voice has also been very familiar. I've not seen the face, but immediately he started speaking, I could match the voice with the face. Let's celebrate him as he comes up. Yes.
Good evening, all. Dr. Crosby is supposed to have done a little introduction of what we're supposed to present today, and then so I will just, fill in and see how all of us benefit. My name is George. Well, I am a molecular biologist. I'm a virologist, actually. I'm trained as a virologist. I work on viruses, and in humans and also in animals. I've worked with non-human primates. My PhD was viruses in bats. And then, well, my expertise is mostly next generation sequencing. That's why you heard me talk about full genome, because that's where the world is going now, right? And I think Africa should also be there. Africa should be there. We are really, really lagging behind. So today, we would try to give us a little just basic of, for example, very few people can still do Sanger sequencing analysis. Very few people, well, thank God for SARS-CoV-2 now, or COVID, that almost every lab now have a PCR machine, because a couple of years back, very few people could even afford a PCR machine, right? But, I mean, SARS-CoV-2 came, almost every lab had to do testing, and that really opened up the space of, molecular, genetics and genomics, which, well, that's some of the positivity of the pandemic. But narrowing in on, I don't know if Dr. Crosby can connect now. Dr. Crosby, he was supposed to be here but had a little emergency. Okay. I think maybe you have to unmute, please, to... So while that is going on, we shared a folder. I don't know if everybody have received the slide, and then there's some of the sequences we're going to analyze. The whole concept of what we're about to do here is, we believe everybody should understand the concept of genomics. In the next 10, 15, 20 years, the world is going to go towards more of genomics. I said something about precision medicine. You're going to go to a hospital, and all they'll do is just do a quick scan, because they already have your genomic makeup. They can check, if there is cancer or if there's anything, and they will give you exact treatment specific to whatever thing that has been seen. That is where the world is going. Okay. Yes, Dr. Crosby.
Hi. Good afternoon, everyone. Good afternoon, Dr. George. Thanks for this opportunity. I want to say hello to everyone in the conference hall, and I am Taiwo Crosby Omotoriogun. I had to apologize for this not being there physically, and I have been able to communicate the reason why I couldn't make it this time right to the conference avenue and for the event generally. So the program, as it's been made available, is that there will be a section that has to do with genomics in general, looking at BAT and putting BAT in perspective. So, but for this case, we plan to look at aspects of genomics, but not the full things in genomics. As you know, this is broad area, and we can only for this event focus on a very small part of it. And as we've planned to do, I think Dr. George has been able to give an overview of what we intend to do, and which is going to be given background of what we want to do, especially if it has to do with DNA barcoding. And of course, I've listened to some of the presentations that dived into DNA barcoding, actually, where they have to use the cytochrome B oxidase subunit 1 as the target gene for identification of this bat species. So this is some of the area I would like to look at before George can continue doing the demonstrations and how to do. So the general overview for today is how you can conceive a project and then establish your objectives and how you go about doing it. And this will put into demonstration. So, Dr. George, what do you expect me to do? Give a background on the DNA barcoding stuff that we have integrated into the plan?
Yes, if you can do that, I think that will really help.
Okay. I don't know how long we have, but I had to swiftly go through the materials that I have with me. If I can share something, I think it will be a good idea.
Yes, please go ahead, please.
So is this helpful that you can see my screen? So, are we able to see what is projected from here, please? I need confirmation for this. Yeah? Okay, please. I'm going to look at a very brief part, but the DNA barcoding, and just to establish this idea, and people can still continue to use and see areas and how molecular identification is possible with this approach. But we will look generally at the case of bats and other animals as well, as we have done before. But first, you all know that bats comprise, if it may have changed now, a lot of things have been ongoing in this area. And you know the composition, the number of species of bat that exists thereabout. But I'm not diving into that, just a way of opening this part to you. And why are bats important? Because maybe they are very good indicator of species. I mean, for ecosystem health, as you know, regions and habitats with conservation values always will have bats in them. And of course, you use bats in assessing the conservation status of this, depending on the species of bat that you could have in such environment. And then provide ecosystem services. As you all know, pollination is there, insect predation, and as well as seed dispersal. These are core services that bats render, and which makes them very valuable. Of course, you could say that about 16% of bat species now are threatened with extinction, and this is because you have a lot of anthropogenic activities that are taking place. And as this is exemplified by BirdLife International 2013, you can see all the categories of activities that currently threatens species globally. And of course, why do we need conservation and management? In this case, if we need this, we had to be careful on how we go about managing and conserving bats, because they can be notoriously challenging, because they can also exhibit some non-tonal behavior and migratory attitudes. And of course, body size, little morphological differences, as people have been trying to tease out in their presentation. And of course, roost disturbances sometimes has something to do here. And the identification problem, which is a major challenge that we're trying to also look at in this presentation. You are aware of morphological assessment. I think I've listened to people who have pointed out sacrificing some of the species, and which may have to do with relying on dental or the palatal ridges, just for identification purposes, and of course the cranial and the baculum features. All of these are some of the ways that taxonomists have used in the past, or still trying to use, to identify species. And I don't know the issues with this. Conservation has gone beyond some of these areas. And then talk about acoustic and vocalization, which we've been aware of, and how different species can produce different songs, different sounds, and this can be used to identify them as well. But then there's the approach of the molecular methods, which is now used. But all of this put together could be methods that can be adopted in studying, surveying bats, and doing all of those research that we intend to do with bats. So I will not dwell on this, but let's look at identification of macroorganisms, which is a very big issue now in ecology. And the two main problems are agreeing about taxonomic units and, of course, recognizing specific organisms. So taxonomy is very key here. And this is where George pointed out in one of his remarks, contribution to what's the boundary. So, I mean, if you're trying to use some sequences, there are parameters that will be produced and which you rely on to make that to distinguish between species. But here, there's the question of what is a species. So it's been very challenging drawing boundaries between species and also subspecies and hybrids. And of course, you are aware of things like reproductive barriers, which automatically is a big thing to contain because you can have species, what you call populations, doing different things at different times of the years and all of that. Even behaviorally, things differ. Synchrony is there. All of these can be put into consideration when you want to identify. And automatically, if you have populations that are not agreeing, then you are talking of subspecies, and of course the case of hybrid as well emerging into the scenario. But how do you handle all of this and how do you tear apart this? So there are programs which will enable you to do all of this, to identify hybrids and things like that. But we can't go into all of these at this particular space that we have in this event.
Sorry, Doc, your slide is not moving,
yeah.
Sorry. Your slides are not really moving, yes. So
yeah. Is this moving?
No, not yet, and we can't control from here.
Okay. Oh, sorry. Can I reproject?
Yes, please.
Okay.
Maybe on slideshow, I think that will make it easier too. We're seeing the slides now. So maybe you can just go ahead and present, but just ensure it's moving.
Okay. So what do you think now?
Yeah, maybe do a slideshow and then that would make it a bit bigger for everyone to see.
Slideshow.
Yeah, I think there's the second icon up, the slideshow, yeah. So that would...
Yeah, I've done that, except you want me to reshare again.
Oh, maybe you just continue. Okay, it's better. It's perfect now.
Is it moving?
Yes, perfect.
Okay, great. So if you permit me to continue from where I stopped, that would be good. If you got my message all the way down to that point. So identification of species, sex, and individual under certain morphological conditions also poses very great challenge because you need to know your species. You need to be able to sex them, and you need to be able to differentiate individuals. So these are relevant areas to note if you had to work with identification of species and things like that. So, but We just have to dive straight to how DNA barcoding has been used in identifying species over time and still ongoing. Now, the idea is making every species count. And of course, to be able to do this, you do not need, you need a diverse expertise in this. And of course, you have people who will be biodiversity scientists, you have people who will be genomic specialists, and you have people who will be technologists, and of course, ethicists. And just as the case had been raised that you need some kind of permission to do work, and depending on the level of research that you want to do. And this is just a representation of the diversity that you can have, but this is not exhaustive, just something to give you an impression of how diversity. And of course, DNA barcoding will want to able to tear apart or have a representative from all of these species, so we can know which one is which in this. So the idea has been that, is it possible that we can have something called a DNA barcoder? The DNA barcoder will just be like a headset, a gadget that is able to, on the go, do DNA barcoding in the feed. So you wouldn't need to collect your samples and go to the lab and go through all of those protocol, DNA extraction, do PCR, do your sequencing and all of that. But right in the feed, you have a gadget. And of course, this is happening now. Things like the Oxford nanopore is able to do exactly what this particular gadget is being predicted to do, even though the Oxford nanopore may be doing something different, but at the genomic level. So the idea is, if you know how it works with your barcoder, when you pick up items in the shop and you have something that scans and tells you immediately what these items are because they have been cataloged in these shops. And then think of that, come from that perspective and look at the DNA barcode elsewhere and that format, where in this case you have the nucleotide bases representing the numbers, which are the things that mainly is put into consideration to identify the organism, the individual, based on where the samples emanated. So here you probably are familiar with what DNA barcode is, and of course you can check. And this is the gene that is harnessed. I beg your pardon, I think they have just taken light from my end here, so you wouldn't see my face clearly. But bear with that. So why is the DNA, the CO1, a very good gene used for
Yeah, sorry about that. Hopefully, we hope we have him coming soon. So while we're waiting on that, slides were shared and then some documents were shared. And I don't know if all of us have received anything. Not yet? Okay, not yet too. So what we wanted this session to be was like, Prof, it's like it has not been shared across. Or can we just walk us through? And so what we wanted was everybody could do like a hands-on of whatever the analysis we're doing with some of the sequences we shared. No, I sent this. So sorry about that. So we sent some sequences that have been sequenced already, cytochrome oxidase 1 and cytochrome B. And we also sent a slide which we hope each person, the goal of this is like to have a hands-on. So bioinformatic analysis, not just doing ecology, but you should be able to like analyze your data, right? Do the sequence, do the blast search, plot your phylogenetic tree, and then you could go ahead to do your Bayesian analysis. So most times you want to look at the phylogeography and phylodynamics of the bat species, right? Compared with other regions. Those are the kind of analysis you need to like make sense of your data. But I think we've been given a very short time. We're hoping we would do some of those phylodynamic and phylogeography analysis. The interesting thing is that you have your GPS coordinates, you have probably the seasonal variation data you got. All of those things would make very good sense. You can even track the migratory pattern, right? Based on combining the genomics and all of those data to tell a bigger story. You see some of these papers in Nature doing all of those things. Africa should be doing it also. But the goal is we're going to start small. I believe everybody here has done PCR. Is there anyone here who has not done PCR before? Is there anyone who has not done PCR, apart from those in social sciences, right? Yeah, so the goal is we start from our PCR, sequence, analyze your data, do the bioinformatics, do your blast search, right? And then plot your phylogenetic tree. That's the goal of what we plan to do probably in, if not today, probably on Thursday. And I don't think there will be time to do the Bayesian analysis. That is the highest, that gives you depth of really understanding your data. With the Bayesian analysis, you can predict the evolution of the different bat species. You can tell when that, you can estimate the evolution time of the bat species. And then with your phylodynamic, you can see the trend of that bat. Is it actually declining? Is the population declining or increasing, right? That's actually, but if we can cover all of that, at least the basic, everybody here should be able to like, you know, analyze some of their sequence data and all of that. But like I think Professor Dr. Crosby tried saying, the goal is doing a whole genome sequencing, just like it has been done for humans. The whole genome gives you the complete picture of the organism. I would always hold that. And then there's everything we do gene is just a fragment. I think like Prof actually mentioned, it's just like a tip of the iceberg. It might not give you the complete picture. I think, like Prof said and others have seen some of the disparities between cytochrome oxidase 1 and cytochrome B. There are sometimes you sequence both, the result will be different. Right? So those are some of the things. But for today, we will just start. I'll try to share my screen on just the basic. What I thought was everybody will have the link, so you just download the app. I want everybody, so genomics should, you know, bat ecologies, especially those in bat research, you are going to be the ones to tell the stories of bats in Africa. And if you can add your morphological data to genomics, good genomics, you're going to have one of the best research. All these papers in Nature, we can do it in Africa. Please, I want to challenge us. Bat genome in Africa is very poor. In Nigeria, I don't think we have up to 1,000 sequences of bats deposited. Do we? For those in the bat ecology. I just said 1,000. I don't think we have up to 1,000 sequences. So these are challenges in Africa. Sorry, the white guys will not come and do it for you. You have to do it yourself. So it's a challenge that they take home from this research. Collaborate. I mean, the Kenyan guys, you're doing so much work. Some of the coronaviruses we have seen, I think most of them Kenyan, and they've done so much bat in, you know, bat study in Kenya, South Africa, right? But Nigeria, thank God for the Adeon Jews and I think Roro, Tianshi, a couple of persons that have done some work in bat, but very few, right? But why I'm trying to say this is genomics is very important. So I would just quickly go directly to my slides and then I'll do the mock analysis. Hopefully when the sequences are shared across us, we should be able to repeat this on Thursday. The goal is that any of us can analyze our data by ourselves, submit in GenBank, and then add that to your paper. It adds beauty to it. Okay, so I'll just quickly, because of time, I think I have less than 1 hour left, right? So I have to present. Yes. Yes, okay, so I'll just continue and then, so this is just a slide. So I tried, let me, good. So these are some of the softwares which I was hoping we could download, and then, because we're going to need some of these things for some of the analysis we're going to do now. And then, like I said, I sent 3 different sequences. I think 2 of them are bat species that have been sequenced, and one of them is actually a virus. So just to have a mix of, for those that are into parasites and bacteria and viruses, and then for the ecologists, how do you analyze all of those, your data? So that's why we had to bring some of those data in. So if you can get the links, these are the softwares which, if you download, that will really help ensure the session is really moving faster. So like I said, I think almost everyone have done PCR, right? And for those that have not done PCR, it's just it's a simple way of amplifying your DNA, right, in a sample to give you enough quantity. PCR is just like the in vitro, in vitro of what goes on in the humans or, other organism. For example, in humans, you have the topoisomerase. Those are enzymes that open up the DNA, right? But in vitro here, you have the denaturation done by heat. So that's why you have denaturation. Sorry, can I use a pointer or something? Yes. So you have the denaturation in humans or in animals. This process is done by topoisomerase or helicase that unwinds the strand. But in PCR, heat opens up the DNA, and when it opens up in humans, you have the RNA primase that lays the primer. But in vitro, you have to put in your primer, right? And then the primer anneals to the opened DNA, which it now anneals. And then after annealing, you have the elongation. The elongation here in humans is done by DNA polymerase. But in PCR, you have the Taq polymerase that does the elongation. And then these copies run millions of thousands or millions of times until you have enough copy that, you can now be able to see. This is just like Amplifix is a tool where you do in silico PCR. We're also supposed to cover primer design and how do you target certain genes to amplify your bat genome to go downstream PCR. But I think because of time, we might not really do that. But for Amplifix, if we have time, I could give you a demo that before you go to the lab to waste your money to run PCR, you could check if your primer is actually working. Because in Africa, we live in a resource-limited environment. You can't afford to waste anything, especially for those who are planning to sequence some of their, bat samples. It would be nice to first run an in silico PCR to check are my primers annealing completely. Most time you run PCR, you can't find anything. So you will run in silico PCR first. It will show you the amplified band. Oh, truly, these bands are amplified. If it works, whatever you amplify here, 99% of the time always works in your wet lab. So this is like doing a dry lab PCR, right? So it gives you a clue and a guide if your assay is working. Hopefully by the end of this session, we should be able to run like a couple of in silico PCR so that before you start buying reagents or your primers, you already have seen different sets of primers and which one can actually amplify the fragment of interest, right? So basically sequencing, there are different forms of sequencing, right? Like I said, what I do presently now is next generation sequencing, where you try to do whole genome sequencing. But the gold standard then was actually Sanger sequencing. And I would not really want to spend so much time here, but we all know the genomic structure of the DNA, right? You have the pentose phosphate, the nitrogenous base, and the OH group. So in the case of the basic difference between the deoxyribo, which is what we have in DNA, and dideoxy, okay, is just a nick here of this OH group. So in dideoxy, during sequencing for the DNA, you have the deoxyribo, the DNTPs that are actually inserted. But in sequencing, we also incorporate the dideoxy. And the dideoxy, the O here, you see, deoxy, you have the OH, but dideoxy, the O is absent, right? When this is incorporated in DNA, it causes what we call chain termination. And that's what you get to see here in your sequencing read. But now the world have gone past this. This is what we're doing like a couple of 50, 40 years back, where you have to run gel, get each of the sequences out. But now we have machines that do all of that. This is what was done previously, right, a couple of years back. But now this machine really helps simplify your work. Okay, it makes the workflow easier. Instead of doing all of this, running the gel to check the direction of sequence. The machine does all of that for you. So as you sequence, put your samples here, it passes through a capillary, right? And when it comes here, there's like an excitation. Remember, we incorporated the dideoxy, and these are all labeled with different probes. Now, when it comes here, there's an excitation. It excites, emits colors, right? And these are what you find here. And so this is what you get, like electrophoregram of some of your sequencing run. You have this kind of picture comes in. So some companies will send this to you, but then depending on where you're sending, I don't know how many of us have a sequencing lab in a school. How many of us? Okay, so most of us send our samples right to sequencing. I do all my sequencing by myself. So, but if you don't have the facility, when I started, we were sending our samples out, right? So some companies would send this PDF file, some will send AB1 file, some will send a text file, right? Depending on what they send. So what do they send to you? Faster. So they already done everything, just send the faster file. So there are some. So for example, when I do my sequencing run, it doesn't give me a faster file. It gives me an AB1 file, so I have to go down, analyze by myself. So what we're going to put us through is assuming you did the sequencing yourself, and the file comes out from the machine. What do you do? Okay? Let's assume the Yibos have not done it for you. You do it yourself from the machine. And what they hope is that every facility should try and have, not just every, there should be a regional sequencing platform, right? Because it's actually very expensive to run a genomic lab, very expensive. Even in Harvard, not every department has, in Harvard they have one like hub where everybody sends their sample to. Okay? In my own lab, we have one big lab center, well, it's also work for the whole of West Africa too, where they send samples for sequencing. So if a school can have, a lab can have one sequencing platform, and then everybody goes there to also patronize, right? That will really help. So the goal here, which after this, I'm just going to jump into the sequences and then begin the analysis, right? So the goal is you have your sequencing data coming in. How do you analyze? That's what we're going to do in the next few minutes. And if time permits, I don't know if we will have the time to plot a tree, right? We might not have the time because it's already late. We're hoping in the next 30 minutes we will finish. So I would stop presenting this. Yes. We can plot a tree. The ministry is moving. Okay, so we will try and see how fast we will go, right, to see. But the goal is not just rushing, but everybody should have an understanding. You have sent your sample, you run your PCR, right, depending on which of the genes you sequence, and then the results came back. What do you do by yourself? Not giving to anybody. How do you do the analysis? That is what we want to do now. So hopefully in 30 minutes, let's see how far we can go, and then, well, if we can plot a tree today, then on Thursday we're going to continue, right? And then plot a tree of probably different bat species. I said something about species demarcation criteria for molecular, for genomics. It is very important. In virology, okay, I'm a virologist, so the ICTV, International Committee on Taxonomy of Viruses, there's a species criteria for every virus. For example, I found 4 new viruses in my part of my PhD work. For me to say it is a new virus, I had to go use the species demarcation criteria to check if my novel viruses met the criteria. That is what should be developed for BAT. I don't know if there's profs, do we have anything like that for genomics? So I think that's one of the things where the Barcode for Life, which Dr. Crosby tried showing us, it's a group of researchers globally, just like GenBank, right? They are also trying to do for all animal species to have a criteria to say so species falls under this category. Just like humans, right? Every human shares 95% genomic makeup. But what makes us different from the Caucasian, what makes the Caucasian different from maybe the Africans and all of that? It's our genetic makeup, and bats have this hidden code that we need to unravel, and that will give us a clear picture. All these ambiguities we're facing with genes will be resolved with that. That's still a virgin land in bat research that needs to be fixed. For me, I think people here should start thinking big. We have done that for viruses. Even though we still find new viruses that can't be classified, but at least there's a threshold that gives you a guide. We will try to explore that threshold. First, we'll explore with blast search. Another way to explore that threshold is to do your phylogenetic tree. Okay? You cluster known species with what you have found and check if the clustering will match in. That also gives you another layer of speciation using genomics. So if I can I proceed now with the hands-on? Are we good? Sorry, I was hoping we can do this together, but let's see how far we would go with this. I'm sorry, I was just running a blast search earlier. Good. So, I think I'll just close all of this and start from scratch. So we have, after sequencing, right, you're going to have a file that comes in this format. For those of you that have installed BioEdit, okay, I'm going to be using BioEdit. I showed in the slides some of the app which you can download, right? So these are some of the app. Amplifix is for your in silico PCR. Mega, most of the people here use Mega, right? But I might not really use Mega. There's BioEdit. So BioEdit helps you trim Sanger sequences. And then Cap3 helps you assemble the forward and reverse into one contig. Emboss Major also helps you assemble your contig. And then Expacit Translate tool is actually to translate the nucleotide to amino acid and then do a blast search. And then APE Plasmid Editor is also one of the app I'm going to use for analysis. So I will try to explore all of this app just to clean my sequences, analyze my sequences, do a blast search, and see what information I get from the sequences. So we did a run, and then some of the results came out like this, right? And then if you have installed BioEdit, automatically this would open and you'll see some of the sequences. So just to have the sequences, you have the forward and the reverse sequences. So the first thing I try to do, let me shift this down. The forward and reverse, I want to trim this sequence. And then there are some things you look out for your sequences. When you have a sequence coming like this, if you check some of these sequences, there are some regions that doesn't look good. There are some regions that look like wobbling. Beginning of region, you see the difference, that this region is different from this region. And then every unique peak shows the sequences you have there. I think for those of us that have done sequencing before, I'll try to be a bit fast here. So these are the primary regions that shouldn't be part of your sequence. So what you do, you trim them off. And you also go to the 3'N. You check if the regions are a bit okay. You see some of these regions too are not too good. So we have to trim this off before we go downstream with our analysis. So in BioEdit, if you're using BioEdit, for those of us that install BioEdit, so when you install BioEdit, okay, you're going to get this icon here. This icon I showed here, this icon here. And then subsequent sequencing, especially your AB1 file coming, you would be able to just click and open in BioEdit. So, yes. It's too fast. And I assume everybody is in the same level. Great. Okay, so I would try to be very slow now. Okay. So first is when you install your BioEdit. So I'm going to be using BioEdit to do this sequence cleaning. In my sequencing platform, I don't have the luxury of getting the text file like other companies send. So what the company have done for you is that they have been able to extract the sequence for you. But in my own, after my sequencing run, this is the result I get. So first, how do I extract my sequence? The AAATTT, which denotes the genome of whatever thing I sequence. When you install BioEdit, like I said, this is the icon for BioEdit, right? So I have installed BioEdit. So whenever the file comes in, it's going to show this icon, and then I will say open with BioID. So subsequently, you would always have this icon. So I have already opened, and then the sequencing came in two forms. All my sequencing, I sequence both the forward and reverse. DNA has two strands. It has the forward strand and the reverse strand. Okay? Most sequencing we do here, we just sequence one strand because of cost, right? Back then when we were sequencing, we're paying $5 per sequence, $5 for reverse. And then for gel purification, $5. Usually you pay $15 per sample. That's if you sequence in macrogen. And for me, macrogen is still, their sequencing run is always very good. I'm not advertising, but most people sequence only one region. Please, I will beg you, if you have the funds, sequence both sides, because it gives you a higher reliability of that region. So what you're doing with Sanger sequencing, sequencing forward and reverse, is what we do with next-gen sequencing. So when you hear next-gen, the sequencing was done like 2,000 times. It has sequenced that region over 100 times, and it gives you a high level of reliability. But what you're trying to do with Sanger sequencing is sequencing just once, twice of the same region. If you have the funds, sequence both the forward and reverse. In this analysis, we sequence the forward and reverse of the same region. So what I'll try to do is, I'm going to open again, right? So just, I'm starting from scratch. So when you double click, it's just going to pop up, and then you have the sequence. But first, how do you extract the raw sequence? So you go to Edit. Under Edit, you have Copy Sequence as Raw Text. So I'm going to copy that and then create a Word document. So I'll just quickly create a new Word document. And then in FASTA format, you always have the icon greater than. And then Control V. So we have the raw sequence. Like someone said, a FASTA format was sent to him. This is what the company did for you, right? And send the FASTA format to you. The FASTA format, all they did was added this greater than sign, and then they gave the label of your sample. For example, sorry. So we'll just call this NG. Sorry, I think I have to do it again. Okay, sorry, I think it's opening a new page. Yeah, so Control V. So in FASTA format, always put the greater than. There are some tools. So for sequencing on blast search and all of that, the FASTA format is like the known format to like read DNA, right? For next generation sequencing data, it doesn't use that. It uses for FASTQ files, it uses a different format. So if you only pick the sequence and do a blast search, you won't get anything until you include this greater than sign here. So we call this NGF for forward. So we have the forward sequence, but we know this forward sequence has primers. Remember, you ran your PCR, you included primers to amplify the known region. You need to trim off the primers from this sequence so that what you have is just the region of interest. To do that here, I always like trimming from the 3' end of the sequence. And please, I would also suggest when you're trimming, you always start from the 3' end. Don't start from the 5' end, okay? To trim. Beautiful question. So, why is that? Not that it's going to do anything, but the tool we were using before, when you trim from the 3' end, it will shift, right, the positioning of the sequence. I don't know if that makes sense. Okay, let me just do a practical illustration, and then I think that will really make sense. So, which region do you trim? You check the regions that are a bit wobbling at the end here. Normally, everything here looks good, but we just want to trim off this little part, right? And how do you know sequences that are good? I think that should be another question. You see, every sequence has a unique peak. Any sequence that you see that this green here corresponds to the green up, the red here correspond to T, the CC blue here, the blue here correspond to CCC, cytosine, right? Just like that, it's color-coded. But there are some sequences that when you open, everything is mixed up. Please, in the name of God, don't work with that sequence, because it is actually a poor sequencing run, and everything you get there will be error. Please, is that okay? There are some runs that come out very poor, but because you've spent so much money, $1 now is like in Nigeria, almost 800, right? And maybe you did so many samples and did like 300 USD, and your result came with all those no peak. You just count your loss and move on. But you might not be able to use that sequence data for anything. That's the sad part of. That's why also wherever you send your samples to a reliable company, because in some case you might not get some of those samples again, right? I think those are some of the things. I'm talking from experience. I'm not saying labs in Nigeria don't sequence well, but I hardly do any of my sequencing in Nigeria. If I don't do it by myself, then some of our collaborators do it. Not that I don't trust the work, but it's because of the experience I had over time that I'm not patronized any lab here. So we're going to go with the sequencing. So I'll trim this. To trim, you hold down and drag. You see it has highlighted the region. Hold down, drag, and then Control C. Go to your Word document. This is the Word document we're working. Just Control Find. It will create like a region. I'm going to make this full. Paste the highlighted region. It has highlighted AAA, but I know that what I highlighted was a 3' N. And I hope when I say 3', 5' N, do we know what that means? Your forward sequence and the reverse strand, right? So the forward, DNA starts with a 5' down to the 3' N. So the tail is like the 3', and the head is like the 5'. I don't know if that makes sense. So I'll remove these last two AA, Control X. I have been able to remove what I don't need from the 3' N. I go back to the same sequence. So I'm going to the 5' N to also see if there are some peak sequences. I think we'll have to do this together. Look at the 5' N. What can you see? Are these sequences making sense? At what region does it start making sense? We're going to do this together. So we'll do the trimming together. At what region? So there's 45, another option. 50, another option. Because we're going to trim together. It's like consensus saying 50. This is very important to ensure the quality of your final result. If you include the primer there, I tell you, it could give you some crazy unspecific result. Somebody says 60. So consensus, what do we go with? 90. 50. Well, if for those of you that want to mice, you don't want to waste cut too much, because you can even cut and start cutting your sequence. Let's try 50 and see what 50 gives us, right? So I will just place the cursor on 50. Remember, I say you drag. Place, hold down. Remember from TT. So I drag to the end. You've highlighted Control C. So I go back to my Word document. I'll just clear this. And then Control V. That's highlighted the region. Does that make sense? So I will just trim off. Delete. So I have been able to trim the forward sequence for trim both the 5' N and 3' N. You have to do the same thing for the reverse strand. Remember I said I sequenced the forward strand and the reverse strand. We'll do the same thing for the reverse strand. So I'll just copy this name. This is NGF. I'll just give this _F. And then copy this to say NGS underscore R for reverse. So I would quickly close this. I'll just close everything. So we go back to the folder and then search for the reverse strand. So this is NG01F, NG01R. That's the reverse. So I double click this. So most times, based on the sequencing run, the peak might not really be high, but with BioEdit, you can raise it up to see how good the peaks are. This is one of the advantages of using this tool. You can also drag down to see, to give you a clear picture of. So, like we did before, we go to the 3' end to check if it's clean, if it's good, and if we can. What do you think about the 3' end? Is it okay? Is it good? Should we trim a little? The green here looks like, I think that's the last sequence, the green here. Is it okay? Yeah, thank you very much. So, are we good, or should we just trim small? What do you think? Let's trim a little. I like that part. So, probably we can trim a little just to be sure that whatever back there is removed. So, we'll trim till the last T. What do you think? Exactly 450, right? And then we cut Control C. So, what did I not do? There's something I have not done. I have not copied the raw sequence. So I can't edit this. The first thing you do after you open BioEdit is copy your raw sequence. So you go again to Edit, copy sequence as raw text, right? You go to your Word document, Control V. So we have the sequence. So I can go back to BioEdit and then Control C, copy the highlighted region. So I would find this Control V, it has highlighted. Remember we said the last region is T, right? So I'll just remove those 4 nucleotides. We do the same thing for the 5' end. So 5', you're going to help me again. What do we trim? What do we leave? What do we go? So look at the sequence again. What have you observed? At what point do we knock off? There's 55, there's 50, 50 again. Where do we go? 50 is winning. Do we go with 50 again? Sorry? You can't see, sorry. Can you see now? 50. So let's try 50 and see. So this TT again, down to the end. Control C. So I'll just find it here. That's highlighted the region we need to trim off. So I'll just highlight that and delete. So we have clean forward and clean reverse, but there is something else we need to do with our reverse strand before we can use it. So when you order your primer, you observe that all your primers are always in the 5', 3' orientation, both the forward and reverse. So am I confusing somebody here? Let's not go too deep. Okay? And most of us are. So what we need to do is, you can't work with the reverse strand as it is. You have to do a reverse complement of this strand. Does that make sense? Am I confusing somebody? You do a reverse complement because this orientation is actually in the 5', 3' orientation. You need to make it balance with the forward strand. You have the coding and non-coding sequence. This run in 5', 3', this run 3', 5'. So you need to revert it to that orientation before you mesh the two sequences as one. So that's what we're going to do now. Please don't merge these two sequences like that without doing a reverse complement. Okay? So I'm going to use APE Plasmid Editor. It's a free-to-use tool. Everything I am showing you is free to use. Please, if there's a way you can save your money from Genius, is good. But if you can find a way to stop spending money on Genius subscription, it will really go a long way, okay? Find free app. I use all my work, including, well, most of them to are doing command line. I try to use free apps, okay? How many of you can, I think Genius license for students is $200, right? Then there's some for institution is 3,000 plus. I don't know how many of you can sustain that every year. So you find free tools that you can do these things without spending. It will really help you. I stopped subscribing. I go free. All the free tools is what I go for. Is that okay? Trip, building, everything, free tools. I'm going to show you a couple of them, right? So let's try and see how we could do a reverse complement of this. So APE, I already have it installed. So I'll try to find it. So this is the icon for APE Plasmid Editor. So I'll just open and then run. So when you install, it's just a Java app you open, automatically it's going to open and then you go ahead and do your thing. So what I'll do, I'll copy this sequence here as it is. Please, you don't copy with the greater than sign. You copy only the sequence. Does that make sense? You copy only the sequence. So Control C, just Control V, okay? And then after you paste it there, all I want to do using this app is do a reverse complement. So you go to Edit. You have so many options there. You have copy, reverse complement, but I like cutting my reverse complement to be sure that I have not left anything there. The cutting gives me assurance that, oh, I have taken it out. What I put in is actually out. So I like, you might copy your reverse complement, you don't need to follow me, but I like cutting my reverse complement. So I cut, remember, look at the orientation of the sequence. It starts with GAT, GT. Can we see? It's too tiny, right? The last sequence is TCC. It's too tiny. So I would try to cut reverse complement. Sorry, I didn't highlight. Control A, and then cut. Good. So I go back here, Control V. So the orientation has changed. So we now, what we're working here is a reverse complement of the sequence. Sorry? Okay, so I'll just Control Z. Look at this sequence. Can you see the sequence better now? Let me not paste, I'll paste this one down. Compare the sequence, what are are they the same? The first one starts with GATG. But the second one starts with GGAC. And the last sequence in the first one is GTCC, the last four sequences. And the last four of this one is CATC. So that tells me that, oh, I have done a reverse complement of the sequence. So I can now comfortably delete this because you're not going to use it for anything. Are we good now? So now you have your clean forward sequence and your clean reverse sequence. So what you do is you match them to form a contig. It is your contiguous sequence. Contiguous is like a consensus sequence that you submit in GenBank. Does that make sense? You submit your contiguous or consensus sequence in GenBank. When we also do full genome, shotgun sequencing gives you small. You match them together to form what you call a scaffold. You assemble the genome. What we are trying to do is assemble this genome. Does that make sense? To assemble a genome, we're going to use a free online tool. Like I said, everything should always be free. We'll go online again to see what tool can we use to assemble this genome into one contig. So I would use CAP3. I really hope that all of us would, get the app and then, so CAP3, just CAP3. So CAP3 sequence assembly program comes up. So I'm just going to open it. So what it does here, you see, enter your sequences in FASTA format or FASTA format, right? So we are going to copy the forward and the reverse in here and just click submit. That's all you're doing. So we go back to our Word document. Control A, Control C. That's I copy everything. So I go to Cap 3, Control V, and click submit. So the run has finished. You have running done, results, you have contigs, you have single sequences, you have assembly details, but my concern is contigs. That's all I want. It has generated one file that is representative of the forward and the reverse strand. This is what you're using going forward. For all your analysis, for tree building, first, we're going to do a blast S section. Let's assume we don't know what this is. Let's assume you got an anonymous sample. So what WHO does in some of the accreditation is they send you an anonymized sample. They know what the sample is. Okay? They want to see if your lab can identify that virus or bacteria or anything. So you would do all of this, do your sequence. If it's culture, you do all of that, sequence, and get your result. And you write to them, this is what we found. So that's the same thing we're trying to do here. So Control A, Control C. I'll paste this in the Word document so I can keep track of this. So we have the contig file. What name did we give it? NG, right? So we'll just say NG contig. You must be consistent with your naming. It's very important that you keep track of your file, right? So we'll just do this NG underscore contig. So the next thing we need to do is do a blast search. I'll spend a little time to give you different flavors of blast. You don't just have blast N, there's blast X, there's blast P. What does each of these blasts do? And how do you interpret a blast result? It is very, very important. So let's copy our contig and get to know what this sequence is. Is it a human gene? Is it a bat gene? Is it a virus gene? Let's get to unravel the puzzle. So we copy this. First, we're going to try to use blast N. So these are the different flavors of blast. Blast N is nucleotide blast. Okay, what it does is that it picks your nucleotide sequence, searches a nucleotide database, and tries to find the closest thing to it, and tells you this is this. Blast P is the protein blast. You can translate this nucleotide sequence to amino acid sequence, and you search using that amino acid sequence to check. Most times, protein analysis is a little bit more robust than nucleotide, because the genetic information is mostly encoded like it's in your proteins. Don't know if I'm making sense. It's a bit more robust. And then you also have the blast X. What the blast X does is that it picks your nucleotide sequences, translates all the sequences to amino acid, and search amino acid database for you. So we'll try to explore some of these and see how we could play around. So I would Control A, Control V, press the sequence, and you don't need to change anything. You can go with the default. Okay, but one of the things I tried, we're using a non-redundant database here, right? So I like show results in a new window when I'm using my blast search. So I just click on search. So let's see what the result will come out. Let me close this one so that it doesn't confuse us. So the run is almost ready. I don't know what this is. So let's see what this sequence is. And then when the result comes, we're going to use that to explain the different interpretation. So in BLAST, you have the E value, you have the percentage identity, you have so many things there. Good, the result is ready. Oh, sorry, I used a BLAST X. This is crazy. Awesome. So BLAST X came out and said, oh, this is cytochrome B gene, human. But the question is, would the result I get from a different BLAST give me something else? Right? So this is what BLAST X has shown. Let's try BLAST N. So I always use more than one BLAST flavor just to be sure. And in some cases, you might be more confused Because some cases, what BLAST-X gives you might be different from what BLAST-N gives you. The question is, what is going on? Okay, so we found a human gene. This is BLAST-X. Saying that, so remember this is the BLAST flavor, BLAST-N. The program is BLAST-N. And then the query was 476 length. And then this is the result it gave. We also checked with BLAST-X. BLAST-X, what BLAST-X did was that it picked the nucleotide sequence, translated them to amino acid, right? And searched amino acid database to know what gene precisely has been amplified. And the results came out, showed that it's cytochrome B gene, right? And then there were some results it gave. Query coverage is 99%. Percentage identity is 96.20%. These are some of the statistics we need to keep at the back of our mind. The two most important statistics which you need to use when you're interpreting your BLAST result is your query coverage. Your query coverage and your percentage identity. There are sometimes you have a query coverage of 0 or let's say 10%, but you have 100% identity. That is a false hit. That's a false hit. Because you don't have enough genome to really show that. The threshold, those were some of the demarcation when you heard me asking some questions. The threshold for you to truly say this is at least 70%, right? In exam, they say 70% is distinction. At least 70% coverage should be captured. And then you add that with your percentage identity, you can now comfortably say this is the region of interest. From one BLAST flavor. This is what BLAST-X is telling us. So we go to BLAST-N, right? What is BLAST-N saying? Does the result from BLAST-N contradict result from BLAST-X? These are, most time, try at least 2 different flavors. Don't just stop at, oh, I did BLAST. Which BLAST flavor? If I'm reviewing your paper, these are some of the things I want to know because sometimes I will want to reproduce what you have done. Sorry, if you're publishing in Science and Nature, some of those guys have done the work you're doing. And some would go back to your data and reproduce the same thing just to be sure that you're trying to tell the correct stories. We've seen that so many times. The science paper we publish in African CDC, the African Genomics SARS-CoV-2, some of those reviewers, some of them developed some of those tools. They had to go back, check. So you don't just publish in those journals and then think you write anything and send there. So you must be sure that whatever data you're presenting is reliable. So you always check. And then sometimes reviewers might just want to make you, especially data coming from Africa, will want to make us look foolish. But if you know your science, come on, you write a rebuttal, this is what we do. You're not insulting the person, but you know your science, you have used at least 2-3 different platforms to validate your result. That gives you a high level of reliability. So that's why you do your homework first before you take it out. So in this other sequence, for the BLAST-N flavor, the query coverage is 100%. And then the percentage identity is 99.58%. When you have this level of reliability, you can now say, oh, this is likely this sequence. So if you're doing your BAT, hopefully by Thursday, we're going to get BAT data that we sequence, right? Do the same analysis. That's why I said, I think we've really spent some of our time. We're going to plot phylogenetic trees with the BAT sequences. We're going to repeat the same thing with BAT sequences. Check what results gave us and then see how we could go downstream. But like I said, don't just say BLAST and then you go. Check that your query coverage is above 70%. And also check that the percentage identity is at least above 90% before you can say it is in a particular BAT species. For now, there is no species demarcation criteria, but I think some researchers in BAT genomics are beginning to devise certain criteria. that the sequence must be at least 90% identical. Because we all share, every human share the same genetic makeup. The only difference between other humans is 5%. Sorry, humans and chimpanzees, sorry. The difference between we and chimpanzees is just 5% of our genetic makeup. We share 95% of the same genome with them. But the 5% is very significant. So that's why you need also a higher threshold to be able to demarcate this species. And that's why you could have some of those challenges when you did, cytochrome oxidase 1, it clustered, maybe a different bat species that clustered with, probably, was it Hipposideros? Which one? Those are some of the challenges you're going to face until there is a species demarcation criteria stated to say that for you to belong in so species, you need to meet this criteria, both morphological-wise and genomic-wise. And that helps in proper identification. I think that should be the take-home for us. As bat researchers in Africa, how do we do this ourselves first? I mean, we can do this and then propose to the outside world that based on the experience of what you have done, this is what we have seen. And most times, cytochrome oxidase 1 will always give a conflicting result with cytochrome B. I have experienced that a couple of times. At least we did over 100 sequencing of the bat species we found. There were some, discrepancies we found with some of those genomes. Some of them tallied, but some we had those discrepancies. Okay? So, I would want to stop this. Do we have questions? Have I been able to confuse you or convince you? Have I confused anybody here? I've confused somebody. Oh, my brother, they say he's been a bit. Yes. Yes, please. Yeah, novels, yes. Yes. Cool. Yeah, so I'll cite example of the new bat coronaviruses we found, okay, I think that was the first report of bat alpha coronavirus. I know Kenya have produced so many coronaviruses, so many, and which is really interesting work they're doing in Kenya. So there's a criteria for novelty. So before I say this is a novel species, you're right, I first did a blast search. I had a query coverage of 80-90%. But percentage identity was less than 40%. First, that told me that there is nothing in GenBank that is closer to my sequence. And then the next question was it as a result of sequencing artifacts? Because if you're sending your sequence to ICTV, so when we find anything new, we send it to ICTV to also check. Okay, we've found new viruses in non-human primates, bats, some in humans too, right? So this is the first thing you do. Your blast S, you check the query coverage. That is significantly higher. The next thing is you check the percentage identity. For example, coronaviruses. The species demarcation criteria for coronaviruses is that for you to be a member of that species, you must have 90% identity in the amino acid sequence. 90%. The viruses we found, the closest thing to any of the coronaviruses were like 60%, 50%, 70%. So that told us that, we are actually stumbling on something interesting. And then the other virus we found a new bat sapo virus. The closest thing to it in GenBank was 30%. So after that, there are also different tools I use. There's what we call species demarcation tool. This tool works on everything. All you need to do is plug in, for example, like the sequences that had those challenges, like the Taradura species and the Hipposideros. You plug into species demarcation tool. And what the tool does is that it aligns the sequences and then does a pairwise alignment. and tells you what is the identity of all the sequences there to each other. And it gives you a plot to show the clustering of each of the sequences. So when I find novel viruses, I pick all the viruses that have been described before on that. I plug them in and say how similar are those old or previously described sequences closer to mine. And when the plot comes, it also confirms what the blast results showed me. I think that would be another tool that the BAT team would also have to adopt. So with that, you can clearly tell how the clustering looked like. And if you put that in any paper, any reviewer seeing will know that you have done your homework. And consistently, there might also be a need to revise some of the naming of these bat species. If you start having full genome sequences of various representative bat species, I tell you there's going to be a complete renaming of some bat species. That should be, I mean, you can do that with collaboration. We have so many novel bat species in Africa that have not been typed. So many. But if we could, it's very expensive to do a full genome of humans and bats. I mean, even bacteria is very expensive. But if you can collaborate, you just need representative. You have museum samples, right? Just try to sequence as much of those museum samples. That would be like the first representative sequence where subsequent analysis will give us a direction. What you're finding in GenBank is because we don't have enough representative sequences from Africa. The database is a bit skewed. Because we don't have enough sequences from Africa, that's why you're finding something else clustering with every other thing. It's not giving you a true picture. Until we start populating the database with our own sequences, that will always keep happening. It is now merging it with something else that have already been typed sequence because they were the first to sequence. And you adapted their primers. Did you design your own primer? You adapted their primers. And those are some of the biases you've also introduced. So the problem is from that design, study design, you already introduced a bias. Until the full genome, you can do it, it does a de novo sequencing. So it doesn't need any primer apart from the adapters you add. It will give you a complete picture of that genome. If you start populating more of those genomes in Africa, I tell you, we will have to, you're going to be finding novel bat species and at least most of them rename. We are sitting on a gold mine. The bat research is actually a gold mine, especially for Africa. I know what I saw working on viruses in bats. It's like there are so many, I think someone mentioned bats have interesting viruses. Yeah. Yes. Can imagine. So there's that skew. Even in Nigeria, sir, it's not just there. I mean, apart from few people working on bats, very few sequences from Nigeria have been deposited in GenBank. Very few. But we have diverse bat species in this country. So for us, the take home is you're sitting on a gold mine. You're sitting on a gold mine. Some of us here might end up doing bat research for the next 20 years. For example, part of the postdoc offer I got is to do more analysis on, viruses in bats, not just doing the sequencing, understanding the immune kinetics. So viruses, apart from they having antibodies to show previous exposure, viruses have a way to downplay the infection such that the virus threshold is low. It's still there. And most pathology you find is yourself attacking your cells, your CD4, CD8 attacking you. If self-elicited adaptive humoral immune response that causes some of the disease we see. No, actually, yeah. So viruses have a way to downplay, modulate this, their CD8, CD4 cell from attacking them. So they tolerate the virus for as long as. The problem is we humans going to encroach into bats. We don't have business going to distort the ecology, going to cut down trees, going to affect the rainforest. We are the ones making these viruses come out, spill over. The viruses, some of them are like normal flora in bats. They are like, they are there. They don't cause any issues on the bat. Ebola, I mean, I found some Marburg virus in some of the samples I sequenced. That's part of what I'm going to do, virus isolation. We also found one Ebola positive in Edolom Helvom. We're also attempting virus isolation. So these things are there. They don't have any issues. But we keep going to encroach. I was told the bats in Ife were there hundreds of years before the university even came into existence. But now they are trying to cut off all the trees, kill the bats and everything. The more you encroach into their habitat, the more spillover will come out. Is it those trading in wildlife? We have a wildlife expert here. Most times you need bushmeat, He does not kill, actually. He's working on One Health, trying to bridge the gap. Okay, but I mean, the way these wild animals are hunted, including bats. There's a survey we did. I mean, if you know what proportion of people in Ife eating bat, I was actually shocked myself. Some of them said they preferred bat meat to, other meat. And the interesting thing, when we're in the field with these guys, the way they handle the bat, not washing their hand, blood spills, some were killing, and over time you keep having this exposure. But there's also a good side to this. The good side is that you should have asked yourself, why did SARS-CoV-2 or COVID not kill so many people in Africa? Because some of us have already been immunized on different coronaviruses. A study was done in Sierra Leone, I don't know if samples were collected from Kenya. So pre-COVID samples, they were able to find evidence of cross-reactivity to SARS-CoV-2. Meaning that we have been exposed to other coronavirus, so it gave us the immunity. So one thing with cross-reactivity is that when you have the antibody, if something new comes, because there's already memory, it downplays the infection. The same thing with bats, because they're already exposed to crazy stuff. When these pathogens come in, the level of attack will be reduced, and they can stay with those things for as long as. So this is a take-home for us. You're sitting on a gold mine. I'm going to end here because I have taken so much time, but I want to stimulate a mind that you should form a network. And listen, you don't know it all. I might be an expert in genomics and viruses, but come on, they are experts in ecology that know 100 times of what I ever know. Rather than go to kill myself in the bush, and I had the experience, I almost died in the bush, and I stopped going. I work with experts, ecologists. So you have a network of multidisciplinary research, and that is the goal Africa should be aiming at. The big guys in Harvard and have seen this, and they have been able to harmonize this. Please form a collaborative team that will help tell the bigger story in Africa. Thank you very much. Okay. So, Dr. Crosby, can you hear us? Sorry, I've stopped sharing.
Thank you, Dr. George. Thank you, Dr. George. So, I don't know, is this? Yeah, yes, do you want me to do something? Is that the end of this section? I can't hear any response, please.
Hello, Dr. Crosby. Can we just have a brief comment from you to the audience before we round up for today?
Okay, thank you very
Thank
much
you.
for this opportunity again. And I really apologize for the break in the transmission while I was trying to deliver something to you a few minutes ago before George came on the podium to continue from somewhere. So I think George has been able to take you through some of the part of this course, I would say, what we're expected to learn in this part of the event. And as he had rightly said, I think the bat research is a virgin field, and I think that we all need to go into it, and we'll definitely need these collaborations to keep things going. So being diverse experts in this area and also participating in this event, we will need this to work together and do things. So this is a good thing, and I believe that we are still available, and we'll keep supporting us with the knowledge we have so far in this area. And George is there, myself, I'm here, and other people who do have expertise in genomics are also there, who can help with the process. So we continue maybe from tomorrow, depending on what is on the program, and then we can start to go through what we have for the other part of this event. So thank you so much, everyone, and for having me on this particular moment. Thank you, George. I see you.
Thank you very much. Just a word before you go.
I'm grateful. Yeah.
Just before you go, we want to kindly request if you can just have a voice over your PowerPoint so it could be circulated to members. We'll be really grateful.
I'll do that. Thank you very much.
Thank you very much, sir.
Yeah.
Can we celebrate the two speakers, both the virtual speaker and the physical speaker? That's Dr. George. Dr. George, you were speaking with mastery. You were speaking with mastery. I didn't know half of what you were talking about because I'm a sociologist. So I was just hearing genomics, and I was hearing new Englishes, and I was asking myself, but one thing I could see was someone who has a grasp of what he was talking about. And then there was passion and fervor when he was speaking about how that we can embrace the BAT project and make it a novel adventure for African research. I want us to once again celebrate these people for their expertise. Thank you very much. So while we're speaking a while ago, we were taking our personal data on our lives, our research, and it was getting interesting before we stopped to start the new session. And as we wait for our dinner, and as we get ready to go and take our rest for today, we're going to round up on that. And we have just a few minutes. The food is on the way, and we wouldn't want to keep us sitting, you know. It's better to keep ourselves busy. So we stopped at the middle, and we're going to continue from here. How many of us have enjoyed today's program?